Hi everyone, hope you all are doing well. Myself Shikha, I'm going to tell you about a topic that is widely used in research areas to improve various crop species. Not only in plants, even it has given a successful result to find out the candidate gene or loci governing the complex trait, for example, disease resistance. So let's start to discuss this topic in detail. So this is the introduction part of the GWAS. GWAS is one of the advances of association mapping where whole genome of an individual is scanned using large number of markers to find out the candidate gene or loci or even the tightly linked markers based on association between the phenotype polymorphism and the genotypic polymorphism present in an individual. You can see this diagram. This diagram represents the main idea about the GWAS analysis. So this is a maize plant. Its genome is just extracted or you can say isolated by using different methods. So what is genome? If an individual is having 2n is equal to 40 chromosome number, then the genome will be 20. The genome is basically the haploid content of the chromosome number present in an individual or you can say cell. So after stacking the genome, its complexity is reduced by getting the DNA fragments. To get the DNA fragments, RE enzyme is used. RE. In Hindi, it is also known as RE, which is mainly used to cut the trees or stem portion of the tree. So it cuts the genome at the particular position and just DNA fragments are collected. तो फ्रैगमेंट्स हम इसलिए भी करते हैं ताकि आपका नेक्स्ट जनरेशन सिक्वेंसिंग जो अब हम फर्दर करेंगे वो आपका इजी हो जाए क्योंकि जीनोम आपके क्या होते हैं बहुत ज़्यादा कॉम्प्लेक्स होता है देयर मिलियंस ऑफ जीन्स आर प्रेजेंट सो टू just to reduce its complexity we make it in the fragments. After getting the DNA fragments, go for the PCR analysis to amplify the DNA fragment, then do the DNA quantification, then go for the DNA filtering. There are other processing methods are there. After that, just do the next generation sequencing, which is mainly the whatever the sequences of the nitrogen bases present in a particular DNA fragment. It can be achieved here. For example, this is a DNA fragment, you can see. So if it is having the sequence like A, adenine, thymine, guanine, cytosine, so see the exact sequence will be derived by using this method. So there are several methods of the next generation sequencing method. The most commonly used in case of the GWAS is the genotyping by sequencing method. Even Illumina is used. So after getting the sequence of a particular gene from different individuals that you have selected for the GWAS analysis or GWAS study. So here you can see that for this loci, for this particular loci, there is variation for the allele, that is the thymine and the adenine. Somewhere it is adenine is located, somewhere thymine is located. To count this variation becomes the common variation. So to calculate the allelic frequency, it just count the number of the individual having the adenine in place of the thymine. Here you can say this individual, this one and this one. These are the three individuals. So allelic frequency will be 3 divided by 10. 10 is the total number of the individuals which have taken for the study. And the most common question comes in the GWAS is that GWAS basically focus the common variation. Here for this SNP you can see there is only cytosine. That is the only one individual that is having the cytosine instead of the guanine. So this is the rare variation. To identify the rare variation, mainly QTL mapping would be more efficient compared to the GWAS. After doing this sequencing and just to and estimating the allelic frequency, we discovered the SNP or particular candidate gene or its loci where on which chromosome it is present. It can be known. Even the markers which are tightly linked to the gene of the interest can be known. For example, you can see in this diagram, this is a chromosome, and for this trait A, these two are the tightly linked markers. Similarly for this trait B, these two are the tightly linked markers. So why we identify the markers? So just I will take example of common day life. If you have to locate a particular flat number, that 24 flat number where it is located. So if you go on like searching like this where the flat number 24 is existing, it will be too much complicated. To get over that, if I would say it is located in Varanasi, and in Varanasi it is located just near to the Assi Ghat. So it would be easy to locate that flat number 24 by using these two benchmarks. Similarly, the markers just makes easier to identify the particular gene of interest which are tightly linked with that gene. So this is the main overall idea about the GWAS. So what's the history behind GWAS? As we know, GWAS is mainly based on the association between the genetic and the phenotypic polymorphism present in a particular population. So this idea of association between the genotype and phenotype was first introduced by Billington in 1956. In 2000, human genome sequencing was almost completed. So, lot of genomic space are available to study the complex traits. So, this sequencing data were utilized and successful GWAS analysis was done for the human disease, that is the myocardial infarction, and it was published by Ozaki in 2002. Later on, in 2005, Klein studied age-related macular degeneration, and now you can say that more than thousands of papers are there which mainly focusing the GWAS application for the study of the various complex traits in humans as well as in plants. In case of plants, especially in maize plant, Bello et al. Utilized this study to gain the information about the oleic content, that is mainly a fatty acid present in the maize kernels, and it has also used in various crops like the rice and wheat for various abiotic and the biotic stress. How GWAS works? I will take example of maize plant to study plant height rate. So first of all, there will be a population with different height, and from this natural population, first of all you have to select genotypes with the tall height. So you will get one group and another group with the short height plant you have to select. So this will form a two groups of having different genotypes. So this one is the first group consisting of the tall heighted genotypes, and another group that is having the short heighted plants or genotypes. So now, after doing the phenotyping, just its genome isolated from the individual genotype, and next generation sequencing is done to study the allelic variation. So you can see here, after doing the sequencing, this two graph representing the allelic frequency on y-axis and the SNP on the x-axis. You can see here for fourth SNP, there is variation in both the group. In the tall-heighted genotypes, adenine is having higher frequency compared to the dwarf-heighted genotypes. So we can conclude that at this SNP4, there is differences, and this difference is related to the plant height. So in this way, GWAS works. Why GWAS more successful in plants as compared to humans? This is mainly because a smaller proportion of the phenotypic variants are revealed in humans because of the family-based studies. What do you mean with the family-based studies? If two parents are there, they are crossed, and their progeny will be like this. If they are having the four progenies, they will further mate with the another partner, and they will have their progenies like this. If you are studying for a particular disease trait, so when you will select the individuals which are having the disease and which are not having the disease, there will be limited genetic diversity because of their correlated ancestry. So very lesser amount of the phenotypic variation are available. In contrast to this, in case of plants, greater amount of the phenotypic variation are available. Even a rare variation become common in large families or population. आपको पता है कि प्लांट्स में के केस में पॉलन कहीं भी किसी भी प्लांट पे जा सकता है. सो देर इज द मोर चांसेस ऑफ द रैंडम मैटिंग. रैंडम मैटिंग होगा तो क्या होगा? देर विल बी द मोर probability of getting the all kinds of the variants. So it is more successful in case of the plants. Hog was better than QTL mapping. As you know, QTL mapping is based on the family-based mapping population, while GWAS is based on the natural mapping population. It means for QTL mapping population, you have to cross the two contrasting parents. That, for example, P1 is crossed with the P2. The P1 and P2, I said it should be contrasting in their traits, for which you have to do the mapping for that trait, when you have to identify the markers linked to that trait and to do the mapping. So, for example, if for leaf blight in maize, P1 parent is disease resistant and the P2 is disease susceptible. So, in this case, the parents are selected and crossed to get the further progenies. For example, F1, then F1 is allowed to random make, then you will get the F2, then F3, then F4, like that you will get the progenies. So after 6 to 7 generations of the random mating, you will get the population which will be consisting of the individuals derived from these two parents. So it requires the multiple generations to generate this mapping population, and also selection of parent is not a easy job. It also needs time, skill. So digital mapping is having such limitations. While in case of the GWAS, we do the phenotyping and genotyping in the natural population. जैसे मैंने कहा था कि अगर 300 इनबिल्ट लाइन्स हैं तो उनको हम एक कॉमन टेस्ट के साथ क्रॉस करवा के आप उससे पॉपुलेशन जनरेट कर सकते हो. ऑल द इंडिविजुअल्स विल बी अनरिलेटेड. इस केस में क्या है, कहीं ना कोई रिलेशन इन पॉपुलेशन के बीच में QTL मैपिंग के केस में कहीं ना कहीं एग्जिस्ट करेगा, बिकॉज ऑल दिस प्रोजेनीज आर डिराइव्ड फ्रॉम अ पर्टिकुलर क्रॉस, बट हियर ऑल द इंडिविजुअल्स विल बी अनरिलेटेड, बट इट शुड बी होमोजीनियस. ज़्यादा हिटोजेनिटी होगा अगर जेनेटिक लोसाई के लिए, तो वो भी फिर आपको एक्यूरेसी को ये बॉटलनेक कॉज करेगा. सो एल्सो अगर मान लो कि पार्टिकुलर ये क्रोमोसोम है और ये इस जीन को आपको आइडेंटिफाई करना था, एंड देयर आर सेवरल मार्कर्स, फॉर एग्जांपल A B C D E F G लाइक दिस. सो बिकॉज ऑफ द रिकॉम्बिनेशन ये मार्कर्स क्या होगा, इट विल बिकम अ स्पार्स. जहाँ पे आपके पास 6 मार्कर्स था फर्स्ट जनरेशन में, it will get reduced to the 2 markers linked to the particular gene of the interest. So in this way there is sparse markers due to recombination. और QTL मैपिंग का ये भी बहुत बड़ा डिमेरिट है कि ये क्रोमोसोम पे पार्टिकुलर लोसाई को या SNP को इंडिकेट नहीं कर सकता है लिंक टू द पर्टिकुलर ट्रेट ऑफ इंटरेस्ट. वाइल GWAS के केस में क्या है कि हम पार्टिकुलर SNP या पर्टिकुलर पोजीशन को भी आइडेंटिफाई कर सकते हैं. सो इट इज मोर रोबस्ट कंपेयर टू द QTL मैपिंग, and obviously it will have the higher resolution power because it will have the all possible kinds of the recombinants in the population, while here it is limited to the genetic diversity present in the parents. So QTL mapping will have lower resolution power. And in case of the GWAS, it mainly studies the association or relationship between the alleles and the binary trait. तो binary trait क्या होता है, कोई भी ऐसा trait जिसका phenotypic वैल्यू जो हो दो में इसको हम लोग मेजर करते हैं. फॉर एग्जाम्पल जैसे डिजीज रेजिस्टेंस हुआ, इधर रेजिस्टेंस होगा या फिर ससेप्टेबल होगा. अगर रेजिस्टेंट प्लांट है तो आप इसको बोल सकते हो कि इट इज हैविंग द वन वैल्यू, इट इज द 0. मान लो ऑन और ऑफ, जैसे कि हम लोग स्विच को क्या करते हैं, इधर ऑन करते हैं या फिर ऑफ करते हैं. सिमिलरली GWAS वैसे ट्रेट्स को स्टडी कर सकते हैं विच आर हैविंग द 2 काइंड्स ऑफ द फिनोटिपिक वैल्यूज. तो QTL मैपिंग के केस में होता है हम वैसे ट्रेट को स्टडी करते हैं जिनका कि कंटिन्यूअस वेरिएशन होता है. जैसे आपने पढ़ा होगा नॉर्मल डिस्ट्रीब्यूशन में, जैसे प्लांट हाइट है, किसी का 5 सेंटीमीटर है, किसी का 10 सेंटीमीटर है, किसी का 12 है, 20 है. तो इस तरह से ये एक पर्टिकुलर नंबर वैल्यू को फॉलो करता हो, तो वैसे ट्रेट्स को हम स्टडी करते हैं. तो ऑब्वियसली इसमें जो वेरिएबल्स होंगे वो बहुत सारे, हम वेरिएबल्स के एसोसिएशन को स्टडी करते हैं. तो हमारा कुछ वेरिएबल जो होता है वो डिपेंडेंट होता है और कुछ जो होता है वो इंडिपेंडेंट वेरिएबल्स होता है. फॉर एग्जांपल एल्ड आपका क्या है, डिपेंडेंट वेरिएबल है, और अदर ट्रेस जैसे कि आपका फ्लावरिंग टाइम ड्यूरेशन हो गया, ASI होता है, जैसे सिल्किंग इंटरवल, प्लांट हाइट, ईयर हाइट, ऑल दिस आर द इंडिपेंडेंट वेरिएबल्स. तो अगर आपको इनके बीच का एसोसिएशन स्टडी करना हो तो QTL मैपिंग जो होता है वो एज्यूम करता है कि इनके बीच का रिलेशनशिप है वो लीनियर है. लीनियर रिलेशनशिप को ये एज्यूम करता है, LINEAR. ठीक है. वाइल इस केस में क्या होता है, GWAS के केस में ये लॉजिस्टिक रिग्रेशन पे बेस्ड होता है. आपने पढ़ा होगा S-shaped कर्व जो होता है. तो मेनली इसमें क्या होता है, जो डिपेंडेंट और जो इंडिपेंडेंट वेरिएबल्स हैं उनके बीच का जो रिलेशनशिप जो एज्यूम करता है वो इट इज बेस्ड ऑन द लॉजिस्टिक. तो लॉजिस्टिक किस में एप्लीकेबल होता है, फॉर द बाइनरी ट्रेट. को भी बाइनरी डिपेंडेंट ट्रेट का जो रिलेशनशिप हुआ अदर इंडिपेंडेंट ट्रेट के साथ, तो उसमें हम लॉजिस्टिक कर्व. तो यहाँ से एक बहुत इंपॉर्टेंट क्वेश्चन बन सकता है कि QTL मैपिंग किस टाइप की रिग्रेशन पे बेस्ड है और GWAS किस टाइप की रिग्रेशन पे बेस्ड है. सो नाउ यू कैन टेल द आंसर. And what do you mean by the regression? It is basically the association between the 2 or more variables. So this is the major difference between the QTL mapping and the GWAS. This flowchart is very much important for the genetics and the plant breeding students. So If we have to study a particular trait in case of the plants or to find out the SNP or candidate gene related to a particular trait, so what will be the procedure or how you will conduct? So this flowchart will help you in doing that. So the first of all, you have to select the population in which you have to conduct the GWAS. So the the population should be homogeneous and independent, means it should not be related by ancestry, and the desirable size should range between 100 to 500. If you take the population size less than 100, then there will be chances of the overestimation of the phenotypic variance, which may give you the false association. If you take more than 500 population size, then there will be difficulty in handling it and also to manage the experimental error. So after selection of the appropriate population, do the phenotyping. The phenotyping should be done in the multi-located trials or multi-seasons or years or and most importantly in replicated trials should be there. So then you will get the phenotypic data. After getting the phenotypic data, do the statistical analysis to estimate mean, components of variance and the Boston's heritability. So because of the multi-location trials and the seasons and the years data, G into E interaction will be high, and this will reduce the accuracy of the GWAS result. To overcome this, calculate the mean value by in the form of the bluff and blues. As we know that the phenotypic data are highly unbalanced data. To overcome this, just calculate the mean value in the form of the bluff and blues. And this Gaussian's heritability is the most important factor that will decide whether to continue the next step or not. If the population will have the low heritability, then don't go for the genotyping. If it ranges between the moderate to high, then do the genotyping. So the next step is the genotyping. Here you will get the sequences of all the DNA fragments, and then you have to compare the loci with allelic differences. Here you can see these two loci are having the allelic differences. After getting all such data, do the genetic diversity through the PCA, principal component analysis, and also assess the population structure, whether the population is having the subpopulation or not. So this is mainly the population structure. पॉपुलेशन इसका अगर मैं इस पे समझाऊं तो पॉपुलेशन वो होता है जिसमें कि all the individuals will can mate with anyone, so such kind of the population are more desirable. लेकिन अगर उसी पॉपुलेशन में कोई एक सब ग्रुप फॉर्म कर ले और वह ग्रुप्स जो होते हैं वो एक दूसरे में ही से ब्रीड करें, वो इनके साथ ना करें, सो उनका जो एलिक इस ग्रुप में जो एलिक डिफरेंसेस होगा और इसमें जो मेन वाले में जो डिफरेंस होगा वो एक दूसरे से वेरी करेगा, तो सच काइंड ऑफ द पॉपुलेशन स्ट्रक्चर will give you the false result. So it can't be ignored. Before doing the association test, it is most important to study the population structure. After that, also study the linkage disequilibrium. लिंकेज डिसइक्विलिब्रियम क्या है? जैसे कोई दो जीन है, although they are not physically linked, but they inherit from the parents to the progenies or the further generations. तो लिंकेज डिसइक्विलिब्रियम का स्टडी बहुत इम्पोर्टेंट होता है. फर्स्ट पॉइंट तो ये है कि by knowing this you can also estimate the number of the markers required for the GWAS study. और ये बहुत इंपॉर्टेंट क्वेश्चन पूछा जाता है कि how many markers you needed or required to study the GWAS. So it is mainly estimated by the using this formula that the genome size divided by LD decay. LD decay. What is this LD decay? तो LD decay आपका होता है लिंकेज डिसइक्विलिब्रियम डिके. जैसे कि क्या होता है, मान लो कि आपके पास ये पार्टिकुलर जीन था, इसके साथ बहुत सारे मार्कर होंगे जो कि इनहेरिट हो रहे हैं, लेकिन रिकॉम्बिनेशन की वजह से क्या होता है, यह आपका जो मार्कर जो था लिंक्ड वो कम होते जाता है टू द पार्टिकुलर जीन ऑफ द इंटरेस्ट. तो ये आपका क्या हुआ, एलडी डिके हुआ. देयर इज डिके इन द लिंकेज डिसइक्विलिब्रियम. So this will decide the number of the markers required. So this is mainly studied through the heat map, and the population structure is studied through the QQ plot. After doing this study, just test for association for the GWAS. After doing this study, just do the GWAS analysis by using the different software such as the Tazel, GenStat, P-Link, R-Gapit, etc. So this association test mainly done or tested by the FDR or the VC method. What is FDR? FDR is the false discovery rate and VC is bond pharaonic correction. So in case of the FDR, the test of association between the marker SN or you can say SNP or trait is estimated for a particular trait independently. While in case of the bond pharaonic correction, this test is done for all the traits simultaneously. So this is the major difference between these two methods. After doing this test, which model you will apply for the GWAS analysis, it is also important and this can be asked. If the population is having the subgroups or subpopulation, then apply the mixed linear model, that is this model. If the population is not having the subpopulations, then go for the GLM model, that is general linear model. If the population in which you have doing the study, it is having the population structure, means subpopulation is there, and also the cryptic or you can say hidden ancestry और cryptic relatedness पे जिसे हम बोलते हैं, अगर वो दोनों present है तो we apply this model MLM, that is the mixed linear model plus kinship. kinship basically relationship को study करता है और relatedness इसको आप बोल सकते हो, and PCA, that is the principal component analysis. This study the population structure. तो ये 3 methods को apply करते हैं हम लोग अगर आपके population में population structure present हो और hidden ancestry present ho. So this is the mainly the brief idea about how to conduct the GWAS study in agriculture related areas. So here it is the list of the softwares which are used for the GWAS analysis. The most commonly used is the Tazel, and these softwares needs to be updated